All right, we're live. Welcome to the 7th hour AP Bio. All right, anyway, so we're just going to call this thing proteins. This is what the final product will look like. So we have lots of things. It's not as bad as you think it is.
That looks like a lot.
It looks like a lot. We have a lot of pictures. We have lots of pictures. So what I want to start with is just what a protein is made out of. It's a polymer of amino acids. So if you link a whole bunch of amino acids together and then you allow them to fold, you'll end up with a protein. Let me zoom this out just a little bit so we can kind of get this whole first section in there. There we go. Okay, so the parts of an amino acid we talked about on Friday after your guys' quiz, but most of us were half asleep, so I don't blame you guys, I was tired too. So we need to kind of recap that. But an amino acid is made up of 5 parts, and every single amino acid will have these 5 parts. There's a carbon in the chest, hydrogen on their head, a carboxyl group, an amino group, and an R group on their feet. That's an amino acid. And remember that their R group on their feet are going to be the only thing that's going to differ between each amino acid. So you have 20 different amino acids that all have 20 different R groups. And their R groups are going to vary in their chemical properties, which allows for a lot of complexity in how these proteins are going to be able to fold. So you produce just millions upon millions of different types of proteins, and they all have unique structures. And that diversity is allowed for by the number of amino acids and the type of interactions that we see between those amino acids that make up the polypeptide, that make up that chain. Okay? So the R groups are really important. They're going to have different chemical properties. The carboxyl and amino groups are going to provide the regions where we end up linking these amino acids together. So you can see this carboxyl group has a free OH on it, and this amino group has 2 free hydrogens on it. And between those ingredients, we can make water. And like we talked about before, if you can make water, then you can link 2 monomers together. And that's what we see down here. So if I wanted to put 2 amino acids together, what I'd see is water forming between the carboxyl group of one amino acid and the amino group of another amino acid. The water forms H2 and O. The water leaves. And we are left with a covalent bond between the carboxyl group of one amino acid and the amino group of another. We call that covalent bond a peptide bond. It gets a special name. But it's just an ordinary, normal covalent bond. It just happens to be between 2 amino acids, so it gets a special name. The peptide bond. Yes, the polymer of amino acids is called a polypeptide, and that is going to be the primary structure of a protein. Primary structure we show just by doing a 1 with a degree sign. That's going to be the primary structure of a protein. Okay. So, that's just the general monomer polymer stuff. So step 1 is getting a long chain of amino acids. Got it. All right. Any questions there? Are we good? Can I slide down?
Yes, sir.
If you have questions, I will answer and I won't make you feel stupid. But if you would have been paying attention, you would have known that. All right. Am I good to slide down? Okay. So it is not enough just to get a chain of amino acids. We need something extra. And this is because proteins rely on their 3-dimensional structure in order to function properly. It looks like a lot, but it's actually just a lot of diagrams. So, relies on their 3-dimensional structure. Because of this, we end up with 4 different levels of protein structure: primary, secondary, tertiary, and quaternary. Each one builds upon the latter. So here's what we see. Primary structure is formed through peptide bonds between those amino acids. It is simply the order that the amino acids are in. Secondary structure occurs through hydrogen bonding between adjacent amino and carboxyl groups on different amino acids. And there's only two different types of structures that we see produced here because there's not really any diversity in those hydrogen bonds. It's just one thing. It's a hydrogen bond between an amino and carboxyl group. So because of that, we end up with two very predictable structures. You could either have a beta-pleated sheet or an alpha helix. An alpha helix is just what it sounds like. It's a helix. Okay, so you can see how this thing has, it's like a coil. Okay, so it's a helix. And it's going to be formed through hydrogen bonds between here and here of neighboring amino acids. And it produces this coil all the way around. So just hydrogen bonding. Amino and carboxyl groups. Nothing to do with the R groups at all. All the R groups in this case would be sticking to the outside, out here, around the spine of these helix. Okay. Or we could have what's called a beta-pleated sheet, which is just kind of a fold. So beta-pleated sheet would look more like this, where we have hydrogen bonding here, and they just fold over each other. Yes. Intermolecular interactions between R groups. So R to R, that stands for R groups. So R group to R group interactions. So what this would look like is if we had an R group here that could interact with an R group here, then they would fold towards each other, for instance. And since we have 20 different types of amino acids, that means we have 20 different types of R groups. And they're all going to be able to interact in different ways. And that's going to provide this really intricate three-dimensional structure that we end up seeing between these things. Those are all phase one on that side. So you just need the three things closest to me. 1, 2, 3. And we'll get this posted for you. Okay. Here, do you want me to zoom in on certain parts, like one at a time?
Yes.
Okay. Where are you at right now? Did we get all this secondary stuff?
No. Oh. Yeah, no, it's not.
Okay, so we're right here. So primary is the amino acid structure, the sequence. Secondary is the hydrogen bonding between the amino and carboxyl groups. That gets us either the alpha helix or the beta-pleated sheet. Did you guys get primary already? Okay. So I'll leave it right there then. Tertiary structure is where we get a finalized finished protein. We're going to get that finished three-dimensional shape, and it is going to be due to interactions between neighboring R groups. So for instance, some R groups are polar, meaning they can hydrogen bond with other R groups. Some R groups are nonpolar, meaning that they're hydrophobic. They're not going to be able to interact with water, and they're going to force the amino acid chain to warp to push that amino acid to the inside of the chain and protect it from the water. Some R groups are going to have positive and negative charges, and they're going to attract each other. Or if we have two R groups that both have a positive charge, they're going to repel each other. And if you combine the sum total of all of these interactions, you'll end up with one finalized, predictable, three-dimensional shape that the entire protein will take. But the diversity is immense because of how many amino acids are in a protein. hundreds to thousands of amino acids in this protein chain, all with different R groups that are interacting differently. So we get this huge complexity and diversity in these structures. And every protein has a very particular structure in order to provide its function. Okay. Can I scoot over to quaternary? So the final level of protein structure is quaternary. Not all proteins have a quaternary structure. This is where you have two or more polypeptides that all have their own tertiary structures interacting with each other. So imagine that I have two polypeptides here, this blue one and this orange one. If they have compatible R groups that allows them to interact with each other, then they could stick together and provide some extra functionality as a total between the whole thing. And this is where we have more complex proteins that are going to be able to perform more than one task and function in two different spots. So hemoglobin, for instance, is made up of 4 protein chains. It's made up of 4 individual polypeptides. All of them have their own tertiary structures, but each subunit is going to be able to perform a slightly different task from the next because they all have their own functionality by themselves. Okay, so that is primary to quaternary. All right, so are we good on the 4 levels of protein structure then? All right, I'm going to slide down then to the idea of denaturation. So we'll start here. Imagine I have this finished protein that has its tertiary structure. If you look here, there is a beta-pleated sheet right here. So let's go ahead and label those just so you can see them. So there is a beta sheet there. You see where it like forms this wrapping. And then the alpha helices are obvious. There's an alpha helix. It's the coils. Okay. But you can see that there's more structure and shape than just those because we have extra 3-dimensional structure generated by that tertiary structure. But there are 2 major ways that you can unfold or break or damage a protein. We call it denaturing. You can either change the pH, so this triangle means change. So you can either change the pH of the environment of the protein, or you can increase the temperature of that protein. Both of those conditions will cause the protein to unfold. Now notice that the protein didn't fall apart. It just unfolded. So primary structure is retained. What we lose is everything after primary. So you lose secondary, tertiary, and quaternary structure if you increase the temperature or change the pH of the protein. And when I say change the pH and increase the temperature, what I mean is you have to change it enough to cause it to denature. So there's going to be kind of these tight little windows that all proteins will operate under. And then I'll go down to the text underneath. So here's 2 important points. One, once a protein is denatured, it's always denatured. It's never going to renature. And we see this. If you cook some meat, what you're doing is denaturing the proteins of that cow or whatever it is that you're eating. If you don't eat your food and it cools off, does it uncook? No. That would be really weird. Like you ordered a burger at, I don't know, Wendy's, and by the time you got home it had cooled off and you have a raw burger again. That would be really strange. We don't see that happening. So once it's denatured, it's always denatured. Also, what's happening is the enzyme is breaking. So whenever an enzyme breaks or it denatures, what's happening is the active site is unfolding. So we're losing the structure of the protein itself. The active site unfolds. And I'll remind you guys what an active site is and all that stuff here in a second. Okay. 2 major points down here. Environmental changes are going to impact secondary through quaternary structure. Primary structure is left untouched, and this is because primary structure is formed through covalent bonds, which are very strong. Increases in temperature would have to be insanely high in order to break these amino acids apart. Higher than anything that we would experience naturally. Changes in pH, same way. You're only going to be able to impact secondary to quaternary structure, meaning that the amino acids are still going to be stuck together. They're just not going to be folded properly anymore. So the difference between primary and then the rest of the structures is that the rest of the structures, secondary through quaternary, are going to involve much weaker interactions than covalent bonds. It's going to work through hydrogen bonds or hydrophobic, hydrophilic interactions where R groups are interacting with water.
Question.
Yes.
So when you're cooking something,
Mhm.
Depending on how long you cook it for, it'll get burnt if you cook it too long.
Yeah.
Does that have to do with it being more denatured?
Yeah, eventually you'll start to just completely unwind these things out. Yeah, so different levels of doneness is referring to how cooked those proteins are. And then also all the other structural components. So when things burn, you're sucking so much of the moisture out of these biological molecules that they begin to char. So you're going to start combusting carbohydrates that are present. As the fat gets removed, there's nothing lubricating it anymore. So it starts to actually char things instead of just losing moisture. All right, are we good down here in this bottom left quadrant? Okay. Let's go to the protein rules next. So all the way down at the bottom, if you just draw a line right underneath these little Pac-Man looking guys, we're going to squeeze this into the bottom right corner. These protein rules, I need you to remember for the rest of the year, because we're going to see them a bunch. And I'm just going to expect you to remember this. If we talk about changes in protein structure, this is an important idea. So, the protein rules. Number 1, proteins get their function from their structure. And from now on, anytime I'm talking about protein structure, I'm going to call it a protein's conformation. Conformation equals structure. Rule number 2. When a substance binds to or unbinds from a protein, its conformation changes, its shape, its structure changes. So binding to or unbinding from a protein changes the shape of the protein. And then rule number 3 goes right along with these. If you change the shape of a protein, a conformational change will also change the function of that protein. This gives most proteins a dual functionality, where depending on what's bound to them, they're able to do slightly different things. And this is also the way that we regulate enzymes. So there's a lot of different metabolic pathways going on in your cells that need to be turned on and turned off depending on what's going on around them. So depending on what substances are available to bind to the proteins, we're going to see different levels of functionality kind of unlocked in those proteins. Or different functions that are just completely shut off and inhibited. It just depends on what's going on in the environment. For instance, your hemoglobin, it's really good at picking up oxygen. But if hemoglobin could only pick up oxygen, would you be able to get oxygen into your cells? It would just grab a hold of it and never let go. And now your muscle cells are never, and your brain cells are never going to get oxygen. So hemoglobin, which is a protein, needs a way of picking up oxygen and then strategically dropping it off. And that's where these changes in structure become really important. So how does hemoglobin know to unload oxygen and now pick up carbon dioxide? And then how does it know to drop off carbon dioxide and pick up more oxygen? Are we good on the protein rules? So structure from function, binding and unbinding changes the structure, which then also changes the function. It all goes together. All right, can I slide up? Okay. Let's go ahead and do inhibition next because I want to make sure that we get through this, and then we'll do enzymes last. Okay. Let's focus here. See how there's like a line right here? Right here? No, I made this part of it. That part was already there. So let's focus above that line. We're just going to zoom in right here. Okay? It says non-competitive inhibition. So we need ways to turn proteins on and off. There's two major ways that this can be done. One is called competitive inhibition, and one is called non-competitive inhibition. The way that non-competitive inhibition works is that I always start with the more complicated one because it makes the next one easy. Oh, where at? Which one?
Under the non-inhibition.
Allosteric. So non-competitive inhibition makes up enzymes that are called allosteric enzymes. Allosteric. And what we see with these enzymes is that not only do they have a spot to bind to whatever you're trying to break down, but they also have a separate site called an allosteric site that is going to be able to be bound with a inhibitor, a non-competitive inhibitor. And the way this works is, let's say that the protein, the enzyme, starts off with this shape, and it wants to break down this substance. What we would see is this substrate would just lock into the enzyme. The enzyme would then facilitate the breaking down of this substrate. However, if there is a non-competitive inhibitor present, it can bind to this allosteric site. According to our protein rules, what happens if something binds to a protein? What does that do to the protein? It changes its shape. Yes. So if the non-competitive inhibitor binds to that allosteric site, that causes an overall shape change in the protein, and now the substrate will no longer fit, and no reaction occurs. That is one way to turn off this protein, to make it not work. Now, if we wanted to turn this protein back on, what would need to happen? I would just need to remove that inhibitor. Unbinding from a protein also changes the shape. It will change it back to its original shape, and now it will work again. So these inhibitors work like on-off switches. They're going to make proteins sometimes work and sometimes not. Do I need to zoom in somewhere? Is it hard to see? Oh, I am too. I'll get this posted though for sure. I'm going to slide over. So we're just looking at this black part at 8 minutes. Okay. So we're just looking right here at this black part above where it says competitive. So non-competitive inhibition is more effective because there's no competition between the substrate and the inhibitor. Yes. Enzyme inhibition. Sorry. So we don't have any kind of competition between the substrate and the inhibitor with non-competitive inhibition. This makes non-competitive inhibition work really well. The other type is competitive inhibition, and we see both models used in the pharmaceutical world, but we see a lot more competitive inhibition used in the pharmaceutical world because we can develop drugs that have shapes and structures that very closely mimic the active sites on the receptors. So almost all of like your antidepressants, for instance, are going to have structures that are very similar to the receptors that you're trying to either block or increase uptake of. And what that allows you to do is you can basically have people take certain doses of these inhibitors, which are going to allow you to kind of fine-tune their nervous system that is regulating these different hormones that are being released, these different neurotransmitters. So like dopamine reuptake blockers, for instance, those all work through competitive inhibition. Can I slide down now to here? Is that okay? All right. So with competitive inhibition, what we see is if this is the active site and this is our inhibitor, you can see that the inhibitor has a very similar shape to the active site. When present, it's going to be able to just literally fill and block that site. Right? It's going to prevent it from being open anymore. And what this does is it just simply blocks the substrate. So in this case, we're not generating a shape change. We're not generating a conformational change that causes the inhibition. Now we're just physically blocking the site from where the substrate was trying to bind. So imagine that this is dopamine, for instance, and we are able to produce this inhibitor which blocks that site where the dopamine would fit. And that prevents the reuptake of that dopamine, which that's one of the primary mechanisms in a lot of antidepressants. Dopamine reuptake inhibitors. All right. Last section. Here we go. You guys ready?
No.
We're moving up. All right. This is all about enzymes. So this is an extension of what we just looked at. You guys have heard of enzymes, though, so I saved this for the end, and this is what we're going to spend the rest of the week on, too. So if we need to pick up with this tomorrow, we will. But enzymes are just proteins. They're specialized proteins, and their job is to speed up chemical reactions. And they speed up chemical reactions by lowering the amount of energy it takes for the chemical reaction to occur. We call them biological chemical catalysts. There's a whole litany of chemical reactions that have to occur to keep you alive. You're essentially an animated, walking, talking sack of chemistry. And these enzymes keep all of those reactions progressing at a rate that will actually keep you alive. Without these enzymes, these reactions do not occur fast enough. In order to be considered an enzyme, these proteins have to fall under two categories. One, they have to speed up chemical reactions, and two, they cannot be consumed within the reaction that they speed up. Meaning that after the enzyme performs the reaction, it can go on and perform again and again and again on different substrates. Another thing we see is that enzymes are highly specific. So one enzyme can only be performing one type of reaction. For instance, if you wanted to break down lactose, which is milk sugar, then you have a very specific protein to do that. We call it lactase. And if you do not produce this enzyme, or don't produce it at high enough amounts, or don't produce a functioning form of lactase, then you are lactose intolerant. You can't break down this milk sugar because there's only one protein that can do it. Yes. Specificity. Yes. Enzyme specificity. One enzyme, one reaction. There's two different models that are used to describe this, the lock and key model and the induced fit model. The induced fit model you guys will get a closer look at tonight in that Bozeman video, but it's the preferred model now. We'll talk more about that later. Yeah, hydrolyzed lactase, so like hydrolysis on lactase. So break lactase, or lactose, not lactase. So the enzyme lactase performs the reaction of the hydrolysis of lactose.
So how do people like become lactose intolerant, like when they weren't before? Like it just happens?
So I don't really know like what can end up triggering that, but that all has to deal with gene expression. So there's a lot of different a lot of different environmental things that can turn genes on and off. And that would be an instance where they're no longer expressing that lactase gene where they might have before. So any kind of allergen that is picked up later in life is typically due to gene expression changes. I'm going to slide down just a tad here. 2 minutes. Oh man, we're going to do this. So here is the enzyme. The enzyme is the protein itself, and it's going to have this particular active site. The substrate is the thing that the enzyme is trying to break down. And what we'll see is that the substrate will fit in with the active site, and then we can generate products based on the activity of that enzyme. And in order to be an enzyme, this thing must still be functional after it's done. We have one thing left, and it has to do with if we were to graph out the function of these enzymes, and we can pick up on that like first thing tomorrow. So right when you guys get in here tomorrow, we'll watch a quick video over protein structure, then we'll recap the this sketch note and add this part in the middle, and then we'll get on to looking at our lab for this week. All right, I'll get this thing posted.